Hey guys, it's Miss Carlson here today to talk to you about genetics. The star of the show is going to be Gregor Mendel and the pea plant. Gregor Mendel is considered the father of genetics for his studies with the pea plants that basically gave us our basic understanding of how heredity works. This is all covered in Chapter 11, and we're going to start off with Section 1 with the work of Gregor Mendel. His experiments showed us a lot of things, and we did define genetics as the study of heredity or the delivery of characteristics from parent to offspring. Fertilization is going to play a key role in this process because our reproductive cells come together to form a new cell or new offspring that receive these traits from their parents. We call specific characteristics traits, kind of interchangeably, but you're going to see that term traits more often than characteristics. And these traits are going to be represented in different ways. For Mendel's experiments with the peas, a couple of traits are shown here. The seeds could be yellow or green, and in this case they're green. You also have the pea pod shape that could be smooth or constricted, and in this case it was smooth. So those are two traits that Mendel studied in his experiments. Now, during his study he did have to perform a controlled experiment because peas can self-pollinate and cause fertilization that way, but in order to know how those traits were being passed on and try to figure out the pattern, he had to control that fertilization. And we'll talk more about that later. Now, hybrids are offspring that result from a cross between true breeding parents with different traits. And the factors, or the chemical factors that are controlling this heredity, are known as genes. So depending on what alleles are within that gene, it's going to control what we actually see on the outside. So multiple forms of a gene are known as alleles, and in this picture here of Labrador retrievers, there are different alleles being carried by these offspring. The chocolate lab is carrying what we call a recessive allele, and we normally designate alleles with letters. And the dominant allele is being carried by this black lab here. So capital B is going to be a dominant allele for black fur color. This guy here. We'll show a lowercase b to represent brown fur. And I'll get to the yellow lab here in a second. This black lab could be carrying a capital B allele for black and a second one from mom or dad. So we got to have two forms of two alleles from each parent, and this black lab could have both. If it does, it is true breeding, it's pure. However, because black is dominant, this black lab could have an allele for black fur and an allele for brown fur. But that brown fur is covered up by the black fur allele because it is dominant. So this would be a hybrid. We have two different forms, okay? Two different alleles for that particular trait. Now, the brown or the chocolate lab must be little b, or carrying a brown fur allele from each parent in order for it to be a chocolate or brown fur lab. So, sounds complex, it's okay. We're going to get a lot of practice with this. But if you're wondering how we get this yellow lab, there's actually an exception to Mendel's basic rules going on here. It's something called recessive epistasis, where we have a masking gene in place that covers up the fur color that puppy should have been. So not exactly the same as albinism, or being albino, but similar. And this puppy would have been chocolate or black. However, this masking gene is actually taking away that fur color. But you would be able to tell what type of lab it would have been, chocolate or brown, by looking at the puppy's nose and lips. Its mucous membranes, and if it's more brown, then it probably would have been a chocolate lab without that masking gene. If it's more black, then it probably would have been a black lab. So kind of an interesting way how things work out. But again, that's an exception. We're going to work with just the basics today. All right, so let's move on. And there's an up-close look at those cute puppies before we do. Okay, so the principle of dominance is what I was just showing you there. Dominant alleles will mask a recessive allele when they are present, usually. There are some exceptions to that rule that we'll get into later. So recessive alleles are exposed usually when the dominant allele is not present, as in the case for the chocolate lab. Here's another scenario showing you brown versus blue eye color. We're using the same letters because we usually use the first letter of the dominant trait to represent the alleles. So we have two capital letter B's representing here for brown being dominant, lowercase letter b representing blue eye color. These are both true breeding parents, so they're going to have brown-eyed offspring. These are both true breeding parents because they both have two blue alleles, and they're going to have offspring with blue eyes. Now, if we take two true breeding parents, big B, crossing with the little b, you end up with that hybrid we were just mentioning. So anytime you see two different letters being shown, capital B, lowercase b, you have a hybrid because we have brown eye color and blue eye color being carried on the gene, but the brown is masking the blue because it's the dominant allele. Okay. So segregation is the separation of these alleles that we're talking about. Okay. And so segregation is just another way to say separation. This happens when gametes or our sex cells are forming. So females, we carry eggs as our sex cells, and males carry sperm as their sex cells. Now, there are several generations that we designate when we go through and try to focus on the passing on of these traits. The parent generation is always going to be designated with a P. Okay? And the first filial generation, which is the offspring of the first set of parents, is going to be designated with a F1. F for filial, 1 for being the first. And filial just means relating to the parent. The second one then would be the F2 generation, and that would be the offspring of the F1. So when we talk about the passing on of these different alleles, you'll be seeing these abbreviations used to represent what set we're talking about. The parent, the first set of offspring from the parent, or the offspring of the offspring, which would be the F2. So 11.2 talks about applying Mendel's principles and primarily talks about probability and the use of Punnett squares in order to predict a genetic cross. So if you ever watch a football game or ask somebody to choose heads or tails, there's a 50% chance it's going to be heads or tails because you only have two choices. 1 out of 2 would be 50%. So the likelihood that an event's going to occur is what we call probability. Now we can represent a probability in a ratio as pictured here, like a 2 to 2, or it could be 3 to 1, whatever the situation may be. But that colon kind of separates how many you're going to get out of the total. Percentages are represented by a percent and also with a fraction. So here I have a picture of a cross between a brown-eyed parent and a blue-eyed parent. The brown is designated by a capital B because it's a dominant trait. And the lowercase letter b represents blue because it is the recessive trait. So this parent is brown-eyed because it has a dominant allele. This parent is blue-eyed because it has both recessive alleles. And when you cross them, you will actually do the following. You'll take the b here and the lowercase b and put them into this quadrant. You'll put this little b, and again the little b from up here will go all the way down, and you'll put them into this quadrant. You'll do the same thing for the far one. Take the capital B and lowercase b, and you'll get this combination. Finally, you'll get lowercase b there, and you'll end up with that combination in that quadrant. So in this case, you get a 2 to 2 ratio out of the 4. 2 of the offspring, or 2 of the options have a chance of being blue or brown, and then 2 of the options have a chance of being blue. That's where we get that 2 to 2 ratio. Or you could say 50% chance of being blue-eyed, 50% chance of being brown-eyed. All right, so what I've been showing you are genotypes and phenotypes. And when we have two identical alleles, or those letters that I've been using to represent the phenotypes, we call that homozygous. If we have two different alleles, we call that heterozygous. So going back here, if I were to have this situation, that is heterozygous because there are two different traits. This situation has two of the same alleles, so it is homozygous. If I had two capital B's, that would also be homozygous. All right, and a phenotype then is your physical trait. Okay, so having blue eyes or seeing the blue eyes physically. The genotype or the genetic makeup would be the letters that we've been using to represent those characteristics. So like little b is the genetic makeup or the genotype for blue eyes. All right, the last thing we're going to talk about that Mendel discovered is independent assortment. And this is when genes for different traits segregate independently during the formation of gametes. So if we looked at more than one trait at a time, we would do that with a dihybrid or a two-factor cross, which is going to look really complex to you right now. In this picture here, we're looking at yellow versus green seed and also wrinkled versus round seed. And what is shown in this dihybrid cross, which you can see is much larger than our initial Punnett square I showed you, each of these traits separates independently from the next. So the fact that the seed is yellow or green is not affecting whether or not it's wrinkled or round. It's carried on a different gene for a different trait. All right, so summary of the principles that we've talked about are that genes determine traits and that some alleles are going to be dominant over others that we call recessive. Here's another example. We have a homozygous dominant situation with this earlobe example. Detached earlobes are dominant. Detached earlobes are dominant, and attached earlobes are recessive. If you have the heterozygous version, that dominant allele is going to mask the recessive, and you'll still have unattached or detached earlobes. And then finally, if you have two recessive alleles, you will get the attached earlobe trait. So each parent contributes to a copy of a gene, and these alleles do segregate when forming gametes. So if I looked at this case, if one parent gave the dominant allele capital E and another parent gave the dominant allele capital E, you would end up with this result. And alleles for different genes usually segregate independently, and that's what I showed you with the dihybrid cross. And we won't get into more detail with that until later because it's kind of complex. We'll stick with the similar Punnett squares for now. All right, well, that wraps up Gregor Mendel and his pea plant. Feel free to go back and replay any section that you didn't understand or didn't have time to copy down notes. And don't forget to form those good questions towards your notes to use as a review later. All right, have a good day, and I'll see you next time.